but I have broken it for this one time to make it work. But for me, there's only two pronunciations of fungi, and that's fungi or fungi, which is what you get in the Oxford Dictionary. So there's one in the soapbox at the moment. Okay, so I somehow Richard talked me into doing this talk. They're not two things we normally see together, but anyway, I did a bit of a think about it. Because really it is, when I talk about passion and awe, it's my passion and the awe that I feel, because I have a lot to do with the beekeeping industry. And I have to note that this is Pollinators Week. So it's a week that is largely celebrating bees of all kinds. And so I suppose it's sort of interesting. I sit on a board, the Wing Bee Foundation board, and they deal a lot with all bees and pollination, and they're doing a lot of activity in the Pollinator Week. And there is actually a photo competition for people who submit photos of baby bees. If you go on their website, they've got some most amazing photos being taken of the diversity. I also sit another hat with the Australian Honeybee Industry Council, and that's the peak body for the honeybee industry, which represents Apis mellifera, the honeybee. And I think bees are very, very interesting. And leaving was before you're doing.
And if you want to try the other.
Sorry?
If you want to try the other clicker.
There, click. Oh, this was working correct. It's got something to do with this, not talking to that. But I could try the other clicker if you think that'll work better. Okay. All right. So I suppose the more I thought about it, I think ecological interactions are multivariate and interconnected rather than consisting in single isolated relationships. That's the beauty and the awe of the living world that we're just a part of. We don't sit at the top of the living world, we're immersed and we immerse into it, we are part of the living world. And the more that we look and the more that we study, the more we actually come to understand how incredibly complex that living world is and how beautiful and how it is really awesome. And I think the more I thought about it, bees really do serve. It's a great proxy to illustrate the complexity of natural world interactions. So I suppose when I'm saying bees, there's something like 20,000 bees in the world, and we've got about 1,500 to 2,000, still discovering, of bees in Australia. And those bees, and about 90% of bees are solitary bees. So they provision their young in some kind of a overwintering place with food, and then they emerge in summer and they're great pollinators. But the social bees, which we have the Apis mellifera, the honeybee, is best known, and then we have the native stingless bees in Australia that also they have just greater complexity. And it's interesting that we know a lot about Apis mellifera. So a lot of the examples I'm going to give you today, when I'm talking about this, will actually be Apis mellifera. And it's pretty obvious why we know so much about it, because we've been associated with Apis mellifera as humans for about 5,000 years. That can go back to records of honey hunting in cave paintings or paintings 5,000 years ago. And we know that it was very, very important in Egyptian times. Honey was actually part of their, it was used, it was as valuable as gold. And honey is a food, and it's also medicinal even in those days. So, and if you think about it, most of the food we eat in this country is, the plant food is of European origin, and the major pollinators of those is the European honeybee. So the European honeybee in this country is extremely important for food production. We have the native stingless bees are also good pollinators, but they don't tend to be able to fly as far. They're a lot smaller. They can only fly up to 500 metres. European honeybee can fly up to 5 kilometres easily. It can be managed. The hives are easier to manage and split and make new ones. So, okay, so I guess I've just got here representing the fungi back there with the yeasts but I'm going to talk a lot about yeast, the yeast form and the mycelia form, which are the two separate morphological forms of fungi. And I have popped in little pictures of some of the native bees, because that's my opportunity to, because I'm not putting lots of pictures of fungi in, because I think you all know about fungi, but the bees is something different for people in this talk. Okay, so looks like it's working. Okay, so some of the things I'll start off, because the interactions can be negative, through diseases, pest mass reproduction, that could be positive through food, housing, pest control, spore dispersal. And then we've got another couple of interesting emerging areas like the gut biomes. It's one of my favourite stories. I'll talk at the end. I didn't talk about it last year. So I'm recycling a little bit more as Cold War paranoia. I just love this story of how organisations, when they get a bit paranoid, can they come up with really ridiculous things, but science can prove them otherwise. Okay, so we're talking about the negatives. So there are several fungi that cause diseases in honeybees, and perhaps the most well-known one of those is chalkbrood, Ascosphera apis. And the spores are, we believe, largely in the hive a lot of the time, but it's a disease of the larvae, not the adults. And it germinates in the larvae and invades and it mummifies, a bit the same as all the other entomopathogenic fungi that I've spent most of my career in the DPI working on. Where the spore germinates, it goes into the larva, grows, proliferates throughout, and then turns it into a mummy. And if you're unlucky enough to be a beekeeper and find it, the first hint is you start to see all these white mummies outside the beehive because the nurse bees clean them out and throw them out. If they're dark, it means they've got sporulation happening. But what is also interesting, and this is a bit about the awe and the beauty of nature, is that Ascosphera apis, it produces its spores, which are these single small little oblong spores. It produces them in spore balls that don't look unlike pollen. So is there something in that? Who knows? We only know what we've investigated, and there's so much that we haven't investigated and we don't know, but I do think it's one of those interesting things. And then we have stone brood. This is another fungal disease of bee larvae, but this one is more of an opportunistic disease. It's not the common one of honeybees, but it does tend to, it's often a sign that there's something really wrong with your hive in terms of humidity, nutrition, etc. The most common, we've got quite a few Aspergillus, Flavus, Parasiticus, and Niger are three of the really common ones, but Flavus is one of the main ones that affects insects. And the first thing is that the larvae start to get a bit fluffy. They turn fluffy as the fungus is invaded them and growing on the outside, and then they start to get this yellowish colour, yellow-green, because that's the common colour of Aspergillus flavus. But yes, so that's another disease, and of course it's stone brood because the larvae are hard, like the chalkbrood are. They're hard because they've been mummified. The third disease caused by fungi, it's actually Microsporidian, and the Microsporidia now belong to the fungal group, the intracellular parasites. Molecularly, they thought they belonged to Protista, but we've known for quite a few years now. They're actually fungus because it's got fungal genome. And I have to say, I read just recently, it's not called Nosema anymore, it's the taxonomists, damn them, have gone and changed the name again. It's something a little bit harder to pronounce, but you're not going to get the beekeeping industry to pick up on that new name. They'll still keep calling it Nosema. So we have two species, Nosema apis, Nosema serrana. It affects adult honeybees, so this isn't one that affects so much to the larvae, but the adult honeybees get it and increases dysentery. So you know you've got it if you look outside your hive, and basically they've got diarrhea and you get these splats all over the front of your hive as the bees are coming back. It tends to be a sign of stressed bees. It could be nutritional stress or some other nutritional to main stress. And the adults tend to die away from the hive. Sometimes if you see a little bee that sort of might be just walking along the ground or something a bit slowly, it looks a bit sad. Often it's got nosema. And so I know people like to pick up and save these bees, but it's already on its way out and it's dying outside of the hive, which is a sensible thing that bees tend to do. If the queens get infected, this is a major problem for your hive because she'll cease laying eggs and die within a few weeks. So your workers will have a reduced lifespan, and you do know if you've got nosema in your hive because you're getting all those splats of the dysentery, the diarrhea in front of your hive. But again, giving them, supporting their nutrition and sometimes requeening is enough to bring that hive back and cure it. Okay, another negative thing. This is an interesting, this is a pest, which I think a negative thing that a fungus can do. This is something, if you don't know about small hive beetle is a pest of bees. It's not a fungus, it's a beetle. It was one of my areas of research for years. It seemed to be part of my destiny and it still is haunting me and not leaving me alone. I still have to keep talking about it to beekeeping groups. So this is what you'll see when you come and see a hive. You'll see this slime on the outside, and you can smell this fermented smell up to 30 metres away from your hive on a nice summer's day. You get closer and this is what you see, and you realise this is silence, the hive's dead. And you've got this slimy mess with dead bees in it running out of the front of your hive. And it's very magically called a slime out. So, and it has that strong fermenting smell. You look inside and you realise that it's just covered in this larvae and all the honeycomb, everything starts to wilt and because this is the honey gets fermented first. And then you'll find that the whole wax breaks down and you just get this great massive slime with these maggots or larvae just completely in it. And it's interesting that this doesn't happen in every country that has small hive beetle, but certainly that we've been blessed with it in Queensland. And it's a major problem in New South Wales now because honeybees also have varroa mite. And they found that the varroa mite and the honey and the small hive beetle are actually working together to really hit the bees down there, well, because they've had varroa along with us, but I suspect this summer we're going to start getting more of this if we have a wet summer. So it's interesting that I worked on this for quite a long time and I lost a few hives of my own to this, so I guess it was a great way of studying it. And I have a theory, and I had a PhD student, I was hoping she would have done this, but she actually sort of got turned off to something else. And I still think it's an important thing. That my theory was that the beetle is can mass reproduce because of a yeast that's in. So if you look at that slime, and if you, in the laboratory, this illustrates it well, that this over here to the far left, that was originally a piece of honeycomb and a piece of brood, put into a plastic bag, spray of water, and a handful of beetles thrown in. And that's what it looked like after 10 to 14 days. That mass of mess that the larvae is swimming in is actually, you put it underneath the microscope, and what you see here is the large triangular shapes, they're pollen. The yellowish is just the honey and wax, or maybe the wax, and then the little blue dots magnified up, they're yeast cells. And that, again, you can see a pollen cell there. So I can't point it out with this because modern technology, the red light doesn't go into the screen. Yeah, so you can see it's predominantly yeast, and when you do some isolations and clean it up, you get this yeast here, which we know is Cotomia.
Big stick there.
Oh, no. In fact, people that go to sleep with it too. Okay, so we've got the slime, and you put it underneath the microscope, you can see this is wax, pollen, which you typically find anything to do in the hive, and the yeast cells budding. Typical yeast bud, and this is Cotomia omeri when it's been purified in its nice form. And investigations of it, we found that if you examine the eggs and there's a mucilage around the eggs when they're laid, you actually can isolate the yeast from it. And we've isolated the Cotomia from the inside the gut of the larvae. We've isolated from inside the gut of the late larvae. We've also isolated inside pupae. And this is with sterilising the outside, and of course we find it in the gut of the adults. So we found that it seems to be a symbiote that's carried, and we're not the only ones, but people overseas have done this too. But my theory was it's always isolated. It seems to germinate, as I watched in the lab, we seem to germinate and grow at the same time the eggs hatched. But if you inoculated just straight pollen patty or honeycomb or brood, anything that we normally could, in the lab put to small hive, get the small hive beetle on, the yeast did not go slimy, it did not grow the way it grows in sort of previous picture. It's only when the larvae are there seems to be something it's adding, there's something happening together. Again, some great questions for honours and PhD students here. Something is added, the yeast stimulates, but what happens is it converts the food source into the slime, which these larvae will eat, because if you did it in a small petri dish, you'll find that there's nothing left but insect frass at the end. So they've eaten all the slime. And why not? Because if that slime is largely yeast is highly nutritious for all organisms. That's why, when you're raising insects in a laboratory, you usually add a lot of yeast lysate and things like that to it. So we know that it's a symbiote, but I found that normally for insects, when they're breeding, they need, like all young, need a protein source to grow. young babies, human babies get lots of protein from milk early on in their diet. Even insects, all animals need protein. If you look at a beehive, a typical 2-box hive, 3-box hive, the only protein is usually in the brood box, which will be the baby bees or stored pollen. The rest of the hive is wax and sugar and honey, which is carbohydrate. But when the small hive beetle and the larvae get going with that yeast, the whole lot gets turned to slime that we know is going to be yeast-like. And it appears as though the larvae then just keep feeding and feeding. So if they couldn't convert that wax and the honey wasn't converted to yeast to feed growing larvae, there'd be a limited food source. But they seem to be able to use the entire, and I think that's what gives them the edge in high humidity in places like northern New South Wales, coastal New South Wales and Queensland, where this beetle is a real problem. So to me, I think this is a case of a fungus that's supporting the mass reproduction of what's become a major pest to honeybees in Queensland and New South Wales. And certainly New South Wales beekeepers had some really sad stories from last year with the number of slime gnats they got. So that's a little bit, and again, it's showing illustrating the really interesting complexity you get between fungi and insects who live in the natural world. Okay, and then we get on to bee bread. So bees collect pollen for protein, amino acid source, and they collect the nectar for carbohydrate. And all bees do like pollen when it's fresh, but you'll store it for winter. And when they store it for winter, certainly the honeybees, they usually preserve it, and they do that with a whole lot of bacteria, and also they use yeast. And yeasts are really handy because flowers are the natural repository of a wide diversity of yeast, because yeasts are actually fungi which are adapted to sugar. They're the sugar fungi, and they tolerate very high levels of sugar. But with some of the native stingless bees, there's been work done in Brazil, and why Brazil? Because they actually do get honey production. They have a wide number, quite a range of native stingless bees that they can cultivate over there. We are starting to go into this area in Australia with Tetragonula carbonaria and Hogginsau, and I think we've got Osteoclebia, but it's a lot smaller. But we still don't know a lot because we haven't had people doing enough research. There's research coming on. I think there's quite a bit study to happen in Western Sydney University now addressing these questions, but they certainly know that in Brazil that the yeasts are actually providing something that the larvae need. So it's giving extra. And given that all fungi are metabiological, their metabolism, they produce really interesting compounds, nutritional compounds, it's not surprising they produce a lot of sterols, like ergothioneine, which I know that some people think are very, very important and shown to be very important in humans. But it seems that some of these things have been shown to be also useful for insect development. And it seems that in the work they've done in Brazil to show that yeast fungi are really important for that, fermenting, changing the pollen, working on the pollen to not only preserve it, but to produce extra compounds, which are really important for the larval development. And I no doubt will probably, when more work done in Australia on our native stingless bees, will find that too. This bee bread, the bacteria, lactobacillus, are very important for our bee bread production in the Apis mellifera, the honeybees. The role of the fungi hasn't really been examined to the same level that it has with the bacteria in them. So there's still more work to be done in that area. And of course, it's really interesting that when you have times where there's a dearth or a sparse pollen, like at the end of summer, beginning of autumn, and sometimes when it's dry, you may have a dearth of pollen, but bees have been observed in collecting spores from rust fungi. I remember someone sending me a photograph of bees on, sorry, I'm having a blank now. It was frangipani rust. And I think it was possibly Hamish, who I think is online right now listening. And I can't find the photographs, but it was really interesting at the time. But it seems to be that, and we can see that the native bees, this is one of our native stingless bees, is collecting honeybee. But they've also found that, interesting that some of the rust are actually the yellow to orange, the colour of pollen. And it seems that some rust also have a response to UV light, and it seems that some of them, when the spores are produced in pycnidia, they will produce a nectar or a sweet sugary reward. So it's as though the rust fungi are actually inducing the bees in to help disperse their spores. And they have become quite concerned that with the myrtle rust, that they've noticed bees coming and collecting that, there's a bit of a worry about them spreading it. I think there has been concern over but it seems that also they seem to get nutrition from the rust, but some work has indicated that they don't, just pure rust spores are not good for bees. They can be toxic, but if they're mixed in with normal pollen, which they may already have a store of, it seems to somehow add. So again, another story that goes a lot deeper of people there to investigate. Okay, and of course, how could we ever forget mead? One of the most ancient alcoholic drinks is honey plus yeast plus water. And wouldn't that be one of our first fermented drinks? Because honey has a very, very low water activity. Bees make it that way so it preserves their nectar. They take all the moisture out, they get it down to about 17% or lower. That's one of the definitions of honey, actually. But if your honey gets water added to it, the yeasts are already going to be there because they're collecting nectar from flowers where the yeasts are already. So there's going to be a diversity of yeasts in a hive, and so it doesn't take much to then see how if honey gets spoiled with a little bit of water in it, it will ferment. And of course, from that we've got mead, which can be 3.5 to 20% alcohol. And this is a picture. I think these are meant to be measuring specific gravity to tell what the alcohol content is in something. My annual little bit of volunteering in the beekeeping industry tends to be at the Eka each year, where I'm on the mead sales. So I'm having to tell this story a lot about mead nowadays. But I also went to was asked to speak at a natural beekeeping meeting in northern New South Wales a couple of months ago. What was really interesting was a chap he's a doctor, medical doctor, and he's developing a whole lot of meads for health. And so he talked a lot about, we've got the methylglens, which are meads that have herbs in them and spices. And it seems to be that some of the meads are really good, they're a really good medium for adding some healing herbs to them. And he's starting to market these. And then melomel is also a form of mead known that is it contains fruit. It's called a melomel. If it is very low alcohol, like 3.5%, it's called a hydromel. And it's interesting, most people probably know this, but the origin of the word honeymoon comes from the medieval practice of when guests came to a wedding, they were expected to bring mead as a gift for the newlyweds. And the newlyweds were given a supply of mead for a month, to which they were to use that, and was hopefully to ensure their fertility. So I suppose that's for regularly having a bit of a drink of mead to relax them, got them the right mood, playful mood. So hopefully they'd sort of do the deed and there'd be production. But I'd like to think what would happen if they drank the entire month's supply of mead in one night. There wouldn't be much happening with fertility there at all. Okay, so moving on to another good thing that happens with fungi. Okay, in Australia, our eucalypt trees are have a habit of, they're long-lived, but they're very prone to infections of heart rot. So we have trees get a lot of hollows, so tree injuries, decay fungi, so your heart rot fungi, things like Ganoderma, Phalinus, Ladioporus, or potentius is really a major heart rot one. So as trees grow, a lot of eucalypts have a habit of just dropping their branches as they grow. That leaves wounds, and the fungi are really good at getting in because fungi spores are in the air all the time. They get in and they're specifically there to rot the heartwood. You know, in a tree, the living cells are on the outside, but inside is dead material that these fungi are actually specialised in. So we end up with all sorts of hollows in old trees, and the big hollows, small hollows, deep hollows inside with small openings, large hollow openings. And as I said, there's a whole range of heart rot fungi do that, and it's very much a characteristic of our native forests. And they produce homes of lots of things, but they also produce hollows in trees that produce bee homes. Really important bee homes for the native stingless bees, you find them a lot because native stingless bees can't control their temperature the way of their brood, the larvae, the way that the European honeybee can. So they need a really well insulated home. So think a nice big eucalypt tree with a little gap into it and a hole inside, cavity inside, is a perfect place to go in to raise their young and to have their hive, because it's insulated. Wood is an incredibly good insulating material. So a nice big thick piece of wood in the centre of it is going to be cool in summer and it's going to be warm in winter. So and that's what they need to maintain. We find that honeybees also have adapted to this. This is a log that's been cut across with the collar in there. And we find that our feral or unmanaged honeybees, because honeybees swarm regularly. So bees are the individual, but bees form a hive called the bee. And the bee is like an individual, it's a superorganism, an individual organism, and it has a desire to replicate each year. And the replication is a swarm. And when bees swarm, it's half the hive goes off to split and to find a new home. And when they do that, the queen, the old queen, the workers actually control the whole mechanism. They bring on a lot of potentially new queens. And the difference between a worker and a queen is simply that the larva gets royal jelly for its entire life. That happens, it switches on epigenetics and it becomes a queen. So it activates the ovaries. So the workers will bring on several queens. They'll slim down the old queen. They'll stop feeding and make her lose weight so she can fly. And the time will come, half the hive, they'll leave a provision with lots and lots of honey and new queens potentially ready to emerge. And then they go out and they'll leave, there'll be signal, they'll leave the hive and then they'll, in a ball or a swarm, and then they'll hang somewhere in a tree. And I'm sure a lot of people see swarms. At that stage, they're not aggressive because most of the workers there have got their bellies full of honey, because that's what's going to keep them surviving until they can set up a new hive. So they fill up their bellies with honey. And it's really a very, very interesting process. When they're in the swarm, they have to then find a home. So I always thought they found the home before they swarm. They don't. According to the research done by a chap called Thomas Seeley in America, who made his last work. Really fascinating work he's done, and what he's found is amazing, but how he did it is even more amazing. So he's found that the bees, which are normally out foraging, the field bees, which are normally cued in to going out and looking for colours and odour, to look for sources of pollen and nectar, all of a sudden, they don't look for those cues, they swap to looking for dark crevices, which is a pretty amazing thing if you think about, well, where the hell is that knowledge stored, okay? The bee brains aren't really huge, the neurological system. So anyway, so they'll go off and they'll look for cavities, they'll look for suitable house sites. And when they do, they've actually got really specific requirements. So that's what Thomas Seeley found. He said that, and this all makes sense. They go out and they're looking for a cavity. They don't want a small cavity. They don't want one too big. They want one about 40 litres. If it's too small, they're going to fill it up really quickly and they'll outgrow their home. If it's too big, it's going to be too difficult to keep that warm and to maintain and fill it up. So they like about 40 litres is what he found. And they can run around and somehow measure that volume and come back and communicate that to the hive. They also like a southern entrance in our, if you're up in the northern hemisphere, and it's a northern entrance in the southern hemisphere. Why? Because that gets maximal sun, it's best for thermoregulation, particularly in winter. So if you're exiting to the north in southern hemisphere, you're actually going to be maximal sun for the day possible. They also like it to be at least about 5 metres above the ground. Why? Because that helps keep predators away. But the other really interesting thing is he found they don't like a very big entrance. Makes sense. If you're going to defend your hive, you don't want a great big hole, do you? So their optimal entrance is about 3.8 centimetres by 3.8 centimetres, which I find really interesting because there is, I read it a lot and because of different things in the beekeeping industry, there seems to be a lot of aggro. Some people become the non-natives against the natives and see European honeybees as invaders, which I find really ironic because the people saying that are humans and we are worse invaders in this country than any other life form. And so how they can arrogantly say that, I don't know. But all the papers written on native honeybee, European honeybees swarming, I haven't found anything to show that they really are taking away nest sites from other animals. And when you think about this, you could find a possum that could squeeze through that, or birds that could squeeze through that. So it's saying to me, there are all sorts of cavities in our eucalypt trees because of the wonder of the fungal diversity in rotting them. And there's probably lots and lots of cavities for all different types of organisms which can be supported. And certainly the numbers I've seen is that possible, I've got it in here, no I haven't, it's got up there. Some of the research done, I think it's done in Sydney University or Western Sydney University, they were looking at the number of feral hives per hectare in native forests, and they found that in some areas it was up to 100, which is pretty high. But that's all going to drop. So people who don't like European honeybees seem to get their own way, thanks to Varroa, and they've already found that in Western Sydney University has been tracking 50 feral hives in their region, their unmanaged hives of European honeybees. They found that's down to, I think, I think it's down to 1. And that's all because of Varroa mite, which I'll talk about a little bit more of that. So anyway, so hollows in trees, eucalypt trees have probably been a great place for the European honeybee to come and adapt. And European honeybees do provide a lot of great pollination services, and you'd be surprised, people are going to start to notice the next 5 years how much incidental pollination you're getting from them in your vegetable gardens and your orchards at home, because they're probably not going to be there in another 5 years unless you get your own hives in. Okay, another positive thing of fungi is the control of honeybee pests, and I'm really just touching on this bacon because this is my area for 20-something years I worked on this. So this is a small hive beetle, a healthy hive beetle that's covered in fungus, covered in Metarhizium, Bavaria, and these are larvae covered in fungi. So it was work I looked at turning the fungus on them. We didn't progress with the adult because the problem became how do you apply it in a hive economically, and doses you might need to kill them would be enough to kill honey. Well, they won't kill a hive, but they'll affect them. The bees have a habit of cleaning up their hives really well. You'd have to put the spores inside a little trap the way that they do with the current chemical. And I couldn't see the value of putting a fungal spore there and having it take longer to live. There wasn't kill. It wasn't the thing. But what we did find is the small hive beetle the larvae must move outside of a hive to pupate in the ground, and then the adult emerges and comes back and finds the hive and goes into it. That's its weak spot, because when it comes into the ground, it's open to attack from all sorts of things, ants and fungi in the soil. So if we get Metarhizium and we treat, these are part of our experiment we did in the very early days, you put a little container in there where you put Metarhizium in there and you put some larvae in there and you just sort of put mesh on top so the adults can't get out. We found a really, really high efficacy in that. We also took that to the next level, but don't know why it didn't go anywhere, because that work was probably because the floods hit Brisbane at the time we're harvesting our second experiment. That was a really good one and affected it, and then there was no more funding, and I got too busy doing other projects as well. But it's still, I think, something that's got great potential for controlling small hive beetle where you don't move hives. And of course, yeast reliant. So this is a good thing that bees do for fungi now, because a lot of those good things fungi do for bees or bad things fungi do for bees. So the yeast rely on bees dispersal. They're found in environments, they're sugar fungi, they're sugar loving, so therefore they're going to be found in the nectar, which is the reward that plants are giving to pollinators to come and visit them and collect their pollen. They get the sugar reward, and the yeast live in those. And of course, so a lot of yeast dispersal is from fungi. They also, and when we go back to the rust, I think that some other fungal spores are actually dispersed by bees as well, or pollinators, should say, bee pollinators. So, okay, so fungal control for Varroa mites. So this is, I guess this is something that's very topical at the moment, because the Varroa is one of the most devastating pests of the honeybee. And until it got here and I had to start, I became a Varroa mite trainer for about 9 months, delivering workshops on everything about the Varroa. I guess I really, really appreciated why it's a big problem. It's a pest. This thing is only about 1 millimetre. So this is on a larval bee. It's on an adult bee. It gets into a hive of adults. It breeds and replicates underneath the capped brood, so you can't see most of the mites in your hive. If you're relying upon seeing mites on the adult bees coming and going from your hive, by the time you see that, your hive's about to die, and the numbers build up and your hive just crashes. It came into the country earlier, but it was found in June 2022. Highly likely that some idiot who was trying to get an advantage was bringing queens in illegally. We sort of know, but proving it is another matter, and then getting someone to prosecute is another matter altogether. But the whole sad thing with varroa, and we've now got the rest of the world, we're the last beekeeping country to get the varroa, and we've got the knowledge and the experience for other countries, and we know that without some form of human intervention, there's about a 1 in 1,000 chance that a beehive will survive, a honeybee hive will survive on its own. And there are a range of chemical control options available. One of the things is the Australian honeybee industry has prided itself on we haven't had to use chemicals. And a lot of people, this is a big game changer. It's anathema to them to have to use chemicals. But even the soft chemicals, like things like oxalic acid, formic acid, they've all got their drawbacks. And there are synthetic chemicals which do a really good job, but they leave residues in the wax. So it's changed beekeeping completely in this country. But if you don't do intervention, you've got to be prepared to lose 999 hives for the one that might survive. Breeding varroa-resistant queens will happen in the future, but you can't import varroa-resistant queens because they're not adapted. And also bees are really weird. Not weird, they're interesting. They found a great way of maintaining varroa because essentially you've only got one individual in a hive is reproducing, right? They found a great way of getting genetic diversity in that hive because the vast number of bees in the hive are females and the workers. But when the queen goes out to mate, and she's a virgin queen, she doesn't mate with one male. She mates with multiple males. And the more drones she mates with, the better that hive will be, the more diverse it'll be. But because she's mating with a large number, the consequence is that if you've got genetics that you've brought in, it's highly likely they'll breed out really quickly and they'll only be maintained in part of the hive. So they outbreed really quickly. So that's why you have to let them adapt with losing lots of hives and they'll adapt to your geographic conditions. Okay, so varroa control with fungi, I've never, of all the biological control stuff I've been involved with, to me, this is probably would be because the chemicals, there's a huge need for it. So I got involved in technically I'm retired, but doing a crap job of it. And I initiated a project about 18 months ago now because I thought I've worked with fungi. It's a great opportunity to pass on my knowledge and skills to another generation. Also, there's some things with fungi, if you don't really understand, you can get some things really wrong really easily. And I'd hate to see someone get a research grant, go out and do the work, and the whole lot fall over because of something really simple they just didn't understand. And this work has been done overseas, and New Zealand made great progress, but theirs fell over because I heard that they only had a set amount of money, and apparently they had problem with contamination of their cultures. They couldn't mass produce them. They relied upon a third party to do that. And they gave them the wrong isolate back. So nothing happened. And then they ran out of money. And I heard that happened somewhere in America as well, but there is a company in America who are trying to commercialise this, but we're better having our own isolates. So there is, all this work has shown that lab investigations, field trials, there's a lot of potential. It's strain dependent, temperature tolerant, and there's a good option for an IPM program because in any IPM program for any agriculture pest, we have an IPM triangle where the last thing at the very top, it should be the highest intervention, and that's usually where your chemicals are. Unfortunately, in the mites, that's one of your very first things you've got to use. And biological sits under that, but we don't have any biocontrol. So we do have a project in Macquarie University that I advise on, where we're actually just starting that. It's a 5-year project. So interesting. Hopefully we crack it, but hopefully we'll get to collaborate with the people in America and New Zealand too to try and speed this one up. But I do think this is where fungi have a huge potential because if we can find an isolate that is more highly virulent to mites, but low virulence to bees, and then of course, because of the difference in size, you're looking at a dosing there, you should have a fairly wide margin of safety in terms of using a fungus to kill them up. Okay, this is a really fairly recent area. In fact, I think gut microbiome work even for humans It's really only been around for barely 5 years, and it's become a very interesting major area that we're starting to realise that our large intestine and the gut microbiome is really important for our health, and it influences our mental health and our physical health, our immunity, and it's influencing a lot of things. And so it's a fairly new area in research, and I know there's a few groups who've been starting to look at examine the microbiota in the honeybee gut. I don't know whether anything's yet started happening in the native stingless bees. I think they've mainly been, I know in Brazil, they've mainly been looking at the ones in the hive in terms of food production and storage. But what they do find is that, of course, this is a honeybee gut, to find you will get different organisms at each section in that. And the different organisms will also change seasonally, because the bees will be bringing different things back seasonally. That's what they have found with some of the work. But they seem to think that they're an integral part of the honeybee gut microbiome alongside the bacteria. They seem to help digestion, immunity, and nutrient cycling, just the way they do in us. But this is an area that is, there's a lot of projects have been carried on recently, and I think it's an area you're going to get a lot more research into, and a really interesting one. So again, another subtle way that fungi are playing a role in the lives of bees. And then, of course, this is my favourite story. No, it's not my favourite story, but it's one of the ones that I think is really interesting. Yellow rain, fungi bees and Cold War paranoia. So back the US accused the Soviet Union in 1981 of supplying this bioweapon, and mycotoxins, particularly your trichothecenes, are really nasty, and they can be absorbed in the skin, and they can have all sorts of horrible effects and kill people in big enough dose. And they, the US accused the Soviet Union of producing this bioweapon that was being used by counterinsurgency in communist Vietnam, Laos and Cambodia. And they had these eyewitnesses of this yellow stuff coming out of planes and helicopters, and it was caused 10,000 people to die and had these most terrible symptoms, etc., etc. And T2 is produced by Fusarium tristinctum, which grows on cereal grains, but you need to grow a lot to get some too. Anyway, it was a very interesting story, but nothing held up. But it was some wonderful investigation, particularly by a Harvard biologist who, initially there were people collected some of these samples, these yellow dots, and said, well, it's full of pollen, and it's not just pollen, it's digested pollen, so clearly came through the bee gut. We think it could be bee poo. And of course, the US Army responded with, well, they just put the bee pollen in there, didn't they? So it sticks to humans better and to disguise it. But then the biologist said, well, this was supposedly produced in the Soviet Union. You'd have to collect a hell of a lot of digested bee pollen from bees and ship back tons to the Soviet Union where they were supposedly producing the trunks of these. So logic does have to play a role. I mean, you go with logic here. And sometimes things don't stack up. They still wouldn't shift. It's still apparently for many, many years later, it even went into one of the history of biological weapons, that they're still saying it's this yellow rain. Anyway, the biologists who did the further exploration, but they found that they couldn't actually find any reliable witnesses to, and I think they did find some mycotoxins, but they found them in parts per million and parts per billion. And the mycotoxin, particularly this one, is actually a background one that is found in Southeast Asia. So some of the data they collected was actually pretty amazing, the story they did on it, but it came to careful investigation, said it was most likely a case of just yellow rain is actually bee poo, when the bees poo out a lot of pollen. And it seems to be a thing that has been recorded in China, and it was called yellow rain. And the Asian honeybees, like Apis dorsata, Apis cerana, red dwarf honeybee, I can't remember its name, Florea, they don't regulate the temperature of their hives the way that Apis mellifera does. So Apis mellifera is able to regulate their brood to keep it around about 34, 35 degrees centigrade. They don't regulate like that. And it seems that people have now started to record that when the Asian honeybees get, they have heat stress, a really hot day, one of the ways they can help control it in their hive is to lose a bit of their weight, and they go out and have this mass pooping of their pollen. which is the yellow spots, if you examine them, have got digested pollen grains in it. So it seems to be a reaction for these Asian honeybees just doing that. And so while it sounded like a great story, and it was a case of the fungus was the bad guy producing, with the help of the evil Soviet Union, producing this nasty mycotoxin that was going to be used in counterinsurgency and killing the Hmong people. There seemed to be no evidence to stack up, but instead it was simply a bit of natural bee digestion system that was helping them thermoregulate. So anyway, so that's, but the US is, I think, is still sticking to their story, even though there's zero evidence. And it was a Harvard biologist gathered all this wonderful story to show that what it really was. And that's just some of the gorgeous native bees that we've got in this country. Or is that, well, this one's not necessarily your Apis mellifera with little native one, but you've got all these wonderful coloured ones. And I love these leaf colours because you see them in your roses and other bushes with these really neat cut things. You realise it's them chomping away to take that back to provision their, provision their home. They'll have somewhere where they'll lay an egg and they'll provision it with food so that the larvae hatches after winter and it's got a food source. Okay, so that's fungi and bees. Anyone want to ask questions? I only said
Yes,
one.
Alice.
Thank you.
Thank you. Yes.
I have the right to say yes or no. I'll answer.
So these varroa mites, the country that they're from, how do bees where they are from handle varroa mites?
Okay, it's one of these countries where they've clearly lost lots of, I suspect, lots of hives. This varroa has been in Europe. Probably what we did was jumped across to honeybees in Korea, in a few Asian countries, several times. In Korea, I think in the late 1950s, and then it moved across Russia with bee movements. So it's been in Eastern Europe and Europe for, 60 years. And so there's been time to adapt. So some countries haven't been using chemicals, and we know that the mites that came into this country are not resistant to any of the chemicals, because some of the chemicals being used in Europe, they overuse them, permethrin and talcopyr, and the mites in EU, in Britain and in America, you can't use these chemicals anymore because they've become resistant. They're a chemical hits one biochemical pathway, which is why resistance happens really quickly. So we know, we're pretty certain it's come from a certain area where they didn't use chemicals, which meant they would have suffered a lot of bee losses over the years. And the molecular work has been done to know a little bit about, you can tell, I don't know, I'm not a molecular biologist, but anyone here who is knows that you put these things and you examine satellites, microsatellites and all sorts of things, you can basically track something down to a variant from a geographical area. So there is knowledge, but they need more to prove something. And then getting something taken to court is another matter altogether. That wasn't a curly question for me at all, actually. Yes.
I have an incredibly basic question, and then maybe a not so basic question. My incredibly basic one is what defines a bee? Because I can see all the species have a lot in common, and with genomes we can easily track them.
Yeah, okay, the Hymenoptera, I'm not an entomologist, I'm a mycologist who just likes killing insects except for bees. I think it's got a lot to do with the wing structure, the mouth parts. It'll be, any entomologist here can help me out, but there would be very clear taxonomic characteristics at that family level that defines the mass of a bee.
My second question would be, when you propose using fungi to control things like the beetles, and I'm assuming there would be other applications as well, maybe in the future, would that change the honey structure? And is that
no, it shouldn't, because the honey has actually got spores in it already, because bees are collecting nectar from trees. There's going to be yeast in it. And the spores don't tend to germinate in honey because honey's got 17% moisture. So that's why honey gets water in it or if you've got a jar of honey and you don't, in a really high humidity in Queensland, you don't keep it sealed up, it will absorb moisture and it will go off, because once the moisture goes, after a while, you've got to get that moisture level up so the fungi can germinate. Yeast tend to germinate at much higher concentration of sugar than other fungi do. But I know that the metarhizium wouldn't germinate. I have mucked around it, but that will be part of the project, is actually defining. And the other thing is fungal spores don't tend to stay around for a long time. They're like a seed. They have a limited viability, and the higher the temperature, the shorter their lifespan. But the case is directed application and mass application, that somewhere in the life cycle we're going to have an effect to drop the population down, but you're not going to have something sitting around for 18 months or 2 years. Like, some of the chemicals are going to have residues, some don't break down for a long time, although a lot of chemicals nowadays do break down for some time. Does that answer this? Yeah. Enough questions now? I've got a hand up. Okay, yeah. And Wayne just.
Someone online has put their hand up.
It's my mouse.
Oh, okay.
I was just wondering if there's any like practical practices that can help with managing varroa, apart from, obviously there's the chemical controls, but I mean, Langstroth is the main approach for keeping bees. Is there like alternative approaches that are better, like warrior hives where you basically draw a new comb all the time?
I can't comment on that. I think there are, I think if anything, some of the current varroa control options are a little bit harder to use than the warrior hives, and that I can't say that with confidence.
Yeah.
But there are things that we know. I mean, there are non-chemical, these mechanical controls that can be used, and one of them is the varroa mite takes about 3 weeks for its life cycle. And if you can, and it can only breed on brood, if you can actually stop your queen producing eggs, if you can cage her up for long enough, you can stop a cycle.
Yeah.
Then you can actually try and treat all the mites on the adult bees. But you can't cage her for too long, because then this buggers up the way that she lays eggs, etc. The other thing you can do is you can use the fact that varroa mite like a drone brood much more than worker brood. They find it a lot more attractive. Why? Because worker bee takes about 21 days to pupate. A drone takes about 24. So that extra time, because the female mite goes into the cell just before it gets capped. She's really sneaky, because honeybees, the nurse bees, provision their larvae with brood food. She sneaks in there and she dives underneath the brood food, the liquid, because she's got this snorkel outside her backside a peritreme that she breathes through. So the nurse bee comes in, checks out the larva, can't see anything, but she's hiding under the brood food, seals it off. Then the larva, then she comes out of the larva, has finished the feeding, and then there's a signal from as it starts to pupate, that she starts laying eggs. She lays 1 egg every 30 hours. The first egg that hatches after 6.7 days is a male. It's an unfertilized egg. And then the next one that hatches is a fertilized egg, is a female. It's insistent, so he mates with her, and he continues mating with each female who then emerges every 30 hours. So imagine if you've got that extra 3 days, there's a few extra mites you can have in that. So the drones is preferred by varroa because they get more bang for their money, because they get a lot more mites through their life cycle. If you put drone comb into a hive, particularly in spring, early summer, you can encourage the queen, because the queen lays an unfertilized egg for drone or a fertilized egg for worker cell, depending on the width of the cell. She measures it with the back legs. Wide, she drops an unfertilized egg in. If it's narrow, she drops the worker egg in. So you can trick her into producing more drone, but then that will cause the varroa to try and lay more in the drone, but you've got to know to get that drone comb out. Otherwise, you're going to have all of a sudden a mite explosion. So that's one mechanical control. The mite don't like high temperature. They don't tolerate. In fact, they prefer 32 to 34 degrees. They prefer it slightly lower, like chalk brood. They prefer lower than the normal brood temperatures. And if people are now examining using these devices where you take a frame of brood out, you take the queen off, the hives up, and you heat them for so many hours to heat it up, and that will take it up to temperature that's comfortable, not going to affect the larvae, but it will kill the mites because if you're taking them too high. That's still quite tricky, and there's a lot of people testing that out, trying to get the data on how to use it properly. So, yeah, there's a few things. See, in the Northern Hemisphere and cold countries, they have a natural brood break every winter. When it's cold, the queen stops laying and they usually pack the hive down in the middle of winter when it's really cold, but we don't have that because we're subtropical. One of the advantages of beekeeping in Queensland is we've got great floral resources and we can beekeep almost all year because there's brood nearly all year. We don't have much of brood break. So that's why Varroa is still a wait and see whether there's something else that we've got that may counteract, but they're worried that brood breaks don't work as well. You've got to force the brood break and you've got to do it carefully so you don't mess up the queen's laying. That long answer to your question.
No, it's good. Thank you. Thank you.
Anybody else want beekeeping 101?
We're all done.
That's it. I think we're done.
Thank you, Di.
Thank you.